Sam McNamara, and this is my group project research paper presentation for week 4. This week we looked at AAV capsid fitness and how different properties of AAV capsids can determine their ability to deliver genes for gene therapy purposes. So the question asked by the paper we looked at this week was, do different properties of AAV capsids make a difference in their ability to effectively deliver genetic material to target cells? The scientists writing this paper hypothesized that sequence changes in the DNA that encodes for these capsid proteins do make a considerable difference in the capsid protein's ability to deliver genetic material. And we agreed with this. It makes sense that changing the DNA of the AAV capsids, which changes the properties of those capsids, will yield changes in the ability of that capsid to deliver genetic material. So the assays and methods the scientists used for this project, so they wanted to look at all kinds of different properties of capsids and determine which properties were most beneficial and most harmful to a capsid's ability to deliver genetic material. So the first thing they did is they generated all single codon mutants of the AAV2 capsid gene. They used the AAV2 gene because it's one of the most common genes used in gene therapy. So they thought that it would be data collected with the AAV2 gene would be generalizable. So they generated all of those mutants. They also generated all single codon insertions and deletions in this gene to look at frame shift mutations. And they used a wild type AAV2 sequence as a positive control and a stop codon substitution as a negative control. So they did this for all 735 positions of the AAV2 gene and used all this info to create what was called a capsid fitness landscape, which looks at all the different properties of different capsid genes and explains which properties are beneficial for gene therapy and which properties make it more difficult for capsids to participate in gene therapy. So results. The researchers found that there are in fact different properties that help or hurt a capsid's ability to participate in gene therapy. So, for example, a mutation to a AAV gene, if the mutation led to an amino acid that is seen in other capsids, that mutation was not as harmful as a mutation to an amino acid that is not in any other AAV. So that's just an example of one thing the scientists discovered. Based on this research, the scientists discovered that an ideal capsid would be one that is mutated to have a negative charge at its internal projections and has also lost a binding site with A20 monoclonal antibody, which is one of the primary antibodies that attacks these capsids and is an issue for gene therapy projects.
So as addressed in the last slide, the researchers were attempting to find which capsid attributes optimize its ability to transfer genes into the cell and basically help put these specific genes that would help treatment options or, serve as treatment options, put those genes into the cell's genome. So the results, the experimentation, proved that various mutations in the cell's coding sequence affects its functioning, as does surface type, like charge, and even a different capsid type. My partner and I completely agreed with the conclusions that some mutations have caused the cell to function worse, and some have caused it to function better and enhance its functioning, but that again comes with certain capsid characteristics that are influenced by genetic modification, such as negatively charged amino acids projecting out of its surface. So that's one characteristic that is crucial to proper optimal capsid functioning. Capsid efficiency is marked by structural features. As I mentioned before, surface charge is one of them, and sequence variability is a big one as well. So the scientists use machine learning algorithms to predict and design specialized beneficial capsids for, again, different treatment options that might be helpful in the medical field. So because of this machine learning technique, people can examine the fitness of a capsid under various conditions, like strenuous conditions that are mentioned on the slideshow. And we thought it would be important to evaluate the reasoning of why some qualities prove to simply be better, and because it would help scientifically engineer artificial or highly specialized capsids to solve these intricate healthcare problems and expand treatment options. So this very much applies to the medical field.